to ask Professor de Voogt to give his presentation. Professor de Voogt works at the environmental chemistry at the University of Amsterdam, and he's principal scientist at the Chemical Water Quality Group of KWR Water Cycle Research Institute.
Good morning. My talk of today is about a specific group of drugs, which are the drugs of abuse. And I would like to give you an overview of the contents of my talk. I will give you some background on why we do this work and the kind of strategy that we apply. And then provide you with an overview of the occurrence of drugs of abuse in surface waters and wastewaters in the Netherlands, including some work on freshwater and drinking water. I would also briefly touch upon a new aspect of this type of research, which is called sewer epidemiology. And I will show you some levels in influence in European cities, influence of wastewater treatment plants, in order to, and the way we use those results for sewer epidemiology. Just to start with an overview of the societal impact of drugs and drugs of abuse. Here you have a list taken from a publication in Lancet some years ago, where you can see some of the drugs of abuse that are very familiar to you. You can see tobacco and alcohol standing in this list, and but the ones that we have been focusing on are the ones that I have circled here: cocaine, barbiturates, benzodiazepines, which are tranquilizers, and as you can see, they have a particular score in terms of toxicity, in terms of dependence, and in terms of societal harm that has been grouped together in order to get to this this ranking of these stimulants. And we are interested in these type of compounds because of the fact that my institute, KWR, is interested in the water cycle as a whole. You probably know that in the Netherlands about 35% of our drinking water is made from surface waters. So we are interested in whether or not, knowing that these compounds actually can occur in surface waters, whether they are removed and whether the remaining quality of the drinking water is such that we that type of compounds don't pose any risk to humans. But of course, once the drinking water has been used in some sort of way, the cycle goes on, and you have additional treatment in wastewater treatment plants. And we also want to know whether these effluents coming from wastewater treatment plants are sources, again, to drinking water, because some of that influent is, of course, discharged from surface waters and used downstream again for making drinking water. And last but not least, perhaps when we have influence and concentrations of drugs of abuse in influence, that may tell us perhaps something about the consumption of these compounds. I'll come back to that later. So which compounds did we select? Well, here's the entire list. This selection is based on legislation. There are formal lists in our legislation called List 1 and List 2. We have monitoring data, both from the societal side as well as from chemical screenings in previous years. Of course, it's also important to avail of standards, and that may sound pretty simple, but it isn't, because obviously as a standard lab you're not allowed to keep cocaine in your laboratory, for instance. So you have to have a license for doing so, and a license means that you have to do a lot of work that can be compared to, for instance, accreditation schemes. well, we try to analyze all these chemicals that are indicated here. Specific techniques are necessary for that. I have listed them in green and red in order to show what kind of mass spectrometry you need. I don't have the time to dwell on the analytical methods too much here, but in principle we concentrate water samples on SPE and then use liquid chromatography and mass spectrometry for the analysis of those. Here you see an overview of the sampling stations that we did in a survey together with RIVM, and the report of that survey is available from these websites. On the right-hand map you can see the locations where we took samples. We took samples from surface waters, we took samples from wastewater treatment plants, we took samples from finished waters, and I'll show you the results in the next couple of slides. In principle, the technique that I used produces results like this. So here you have an example of a typical type of chromatograms that you obtain from in this case, an effluent of the sewage treatment plant in Utrecht. And typically we're talking about concentrations from, let's say, nanograms per liter up to, well, maybe 1 microgram per liter is typically the type of concentrations of tranquilizers that you can find in influents, for instance. Here's an overview of the results of surface waters and drinking water from that report, and I would like to draw your attention to concentration ranges that are indicated here. In surface waters, in raw drinking water, that is water taken from surface waters and treated initially, and then in some finished waters. And as you can see immediately from this graph, in finished waters we normally don't find any of the compounds anymore, except for some barbiturates, which are found at levels of about, say, 1 to 10 nanograms per liter. These are compounds with a very high polarity, and that's why they are very difficult to remove, obviously, by the treatment steps used in our current drinking water treatment. What you can also see is that in surface waters, from the percentages given here, that quite a few of these compounds abide in these surface waters. So in principle, you can find quite a few of those compounds to be present in the surface waters that we have sampled. You can use these compounds to estimate loads. Loads discharged by rivers, for instance. So by simply using the data analyzed in the river, for instance, the River Rhine, where it enters the Netherlands, or the River Meuse, where it enters the Netherlands from the south, you can calculate the amount of drugs coming in kilograms per year. And as you can see, for these groups of compounds, we have minor contributions from the River Meuse and major contributions from the River Rhine. Of course, the River Rhine has a much larger flow rate than the River Meuse, which is the major cause for this difference. But what you can also see is that going downstream in the Netherlands, we have an additional amount of, or an additional load of drugs being added to the system. Not surprisingly, of course, because why would upstream drugs of abuse being generated between brackets into the river, and why wouldn't then the downstream area not generate to that? But this means that there are obviously sources of these compounds also within our country, and that's why we need to know, are they going to be removed in certain treatment facilities. Here's an overview of the results we found in wastewaters. And if you look into the gray columns, I hope they are visible in the back. The gray columns again tell you concentrations of the various type of drugs of abuse in influents and in effluents. And as you can see, for quite a few compounds, the treatment in the sewage treatment plants is such that it does remove the drug of abuse, but since we don't have zeros here, it means that there's still quite a bit of say discharge of these drugs of abuse through the influent in our receiving surface waters. There are also examples, for instance, where we see that the effluent has similar concentrations as the influent, which means that there is no removal at all. Even in some cases, you can see that there is actually no compound present, obviously, in the influent, whereas there is some in the effluent. Now, this may be caused by two different things. Sometimes our limits of detection in influents are higher than in effluents because of the more complicated matrix. But sometimes you also may have deconjugation in the sewage treatment plants of conjugates formed in the body, and that's why once they're being deconjugated, they will be analyzed, of course, in the medium where after the deconjugation took place. So the general message is that although there is some removal in sewage treatment plants, the removal is not sufficient such that we don't have point source, that these STPs form point sources to the surface waters. Oh. So in influence, we can see that when we look at the 8 different sewage treatment plants that we sampled in the Netherlands, we find throughout the Netherlands, we find more or less similar concentrations in the influents themselves. Of course, that doesn't mean that the use of these compounds has been the same in the population areas that were covered by these sewage treatment plants. But as you can see, in general, you find concentrations of benzoyl agonine, which is the major metabolite of cocaine, and of oxazepam, which are close to 1 microgram per liter in influence. I still have to get used to using this machine. In effluents, to summarize, we actually observed 24 of the 30 compounds that we tried to detect. And again, the major ones I've shown you already in the previous slides are compounds like the tranquilizers, the diazepines, benzoyl agonine, the metabolite of cocaine. And there's also a couple of compounds that we obviously do not find. So that brings me to sewer epidemiology, which is more or less, I could say, a new area of research. And that sewer epidemiology is based on the idea that you may use concentrations that you observe in influents to back calculate how much of the particular compound you're looking at is used in the population that is connected to that sewer. And here you see the basis for that calculation. In principle, you use the concentrations themselves and multiply them with the volume of the wastewater inflow in order to get the daily load. And if you then know the amount of people connected to that sewer, you can actually calculate the daily load per, say, 1,000 inhabitants. And if you know the excretion rates of the particular drugs of abuse, then you can actually calculate how much of consumption is involved in order to produce that daily load. So by doing this, you can back calculate the total load shown here for cocaine towards the sewage treatment plant in grams per day. And you can also try to estimate the cocaine consumption. And we've done so on the left-hand axis. Here you see the total load in grams per day that comes into the sewage treatment plant in different cities in the Netherlands, reflected by the bars. And the back-calculated estimated cocaine consumption in these cities is given by the black diamonds. And as you can see, and if you focus a little bit on the estimation figures, you can see that for the survey that we did in 2009, it seems like there's a more or less average kind of cocaine consumption in some of the, say, intermediate towns in the Netherlands. In Amsterdam, the estimated cocaine consumption is almost twice that level, and in some more rural areas you see that the cocaine consumption is less. So this is based on one particular survey using just grab samples from one 24-hour sample from a sewage treatment plant of all these 8 plants. In order to get some more information of this kind, we did an additional type of monitoring by monitoring the sewage treatment plant influence from in total 5 sewage treatment plants in the Netherlands during a whole week, collecting 24-hour samples every day during that week. And what you can see is that for some drugs, not surprisingly, you see increased uses during the weekends. And for other compounds, you didn't see that particular increase during weekends. For instance, for cannabis, there is usually no specific increase during weekends, whereas for cocaine and for ecstasy, there is. By using these data, you get a much better impression of the use than using just a 24-hour sample, obviously. And here you see some data from that week-long monitoring exercise. And again, you find the concentrations in influents that are sometimes the same as the concentrations in effluents. Again, what is striking from this graph is that most of these, well, quite a few of these compounds are really present in the influents. So if you were not convinced that these are being used, well, this actually shows you that by chemical analysis you can obtain data from the use, and that's the rationale behind the sewer epidemiology, that are probably of similar quality or even better quality than the data obtained so far on use numbers or consumption numbers in different countries that are based on inquiries and questionnaires. We also participated in a study that looked into the same kind of data in 19 different cities in Europe. The number says 16 here, but overall we collected 19 of those. And here are some data from that exercise. They are very recently being generated. Again, the cocaine consumption is depicted here in milligrams per day per 1,000 inhabitants. And here you have the various cities all across Europe. And again, you can see that in Amsterdam, there's a relatively high consumption. Similar holds for Antwerp in Belgium. And this was the basis of a press release a couple of months ago. We did that obviously also for other compounds. Here you see the graph for amphetamine and methamphetamine. In this case, we have the loads, not the consumption, but the loads to the STP, milligrams per day per 1,000 inhabitants. And what is striking is that you can actually see quite big differences between towns in this case, because in some towns there is no use of, for instance, methamphetamine, whereas there is a lot of use of amphetamine, and vice versa. Whereas in other towns you see an almost similar kind of distribution between the two in terms of loads to the sewage treatment plants. This is probably caused by the availability of these types of drugs on the local markets. Similar picture for cannabis. Again, you see some striking difference between large cities, and we also have a picture for ecstasy, MDMA, and here you see also big differences between the various towns. There was actually one very striking number here, which was for the town of Utrecht, and because of that we investigated the data from the week-long monitoring that we had done ourselves in Utrecht a year before these data were generated. And this was actually the picture for Utrecht for the 2 weeks that we monitored. So we had a week monitoring during monitored during 2010 and a week monitored during 2011. This was the picture from 2010, and this was the picture from 2011. And here you see the weekdays. And of course, this is a very uncommon pattern for this type of compounds. So we thought that this was probably due to some illegal dumping of the compounds. And actually, we found out that one day before we had started with our sampling, totally by chance, there had been a raid by the Utrecht police into a house, and they found ecstasy ingredients within that house, but they couldn't find any of the pills themselves, so probably they had been discharged. Here you see the location of the sewage treatment plant that we sampled one day after this raid was done. Here's the actual house, and some people were arrested because of this. And well, the distance could actually explain why we would found the type of profile that I showed you in the previous slide. So with high concentrations and constantly decreasing. So I can conclude that the drugs of abuse that we have monitored, tried to monitor, we can say that quite a few of them are present in Dutch waters, and this is confirmed in other surface waters all over Europe. So we found 20 out of the 30 that we detected in surface waters, with concentration generally in the several tens of nanograms per liter. For some compounds like the benzodiazepines, you can go up as high as a microgram per liter levels. Fortunately, most of them are being removed in drinking water treatment processes, and we found only traces of barbiturates that are far below the actual concentration that may pose any harm to humans. We can also say that we should keep an eye on these compounds, because since the very reason that they're present may of course also cause some problems to aquatic ecosystems themselves. We don't know very much about the toxicity of these compounds to aquatic systems, and we have to be aware of the fact that they are present in our source water, so that we have to be sure that they are indeed as well removed as was shown in the drinking water plants that we monitored. Finally, I think I can say that sewer epidemiology poses some new opportunities. I mean, one of the previous talkers was talking about pharmaceuticals and personal care products, and indeed people have asked me, could we, by analyzing influent data, obtain some information about, for instance, habits in a certain population? And the answer is, of course, yes, we can, but that's averaged across a whole population. And we cannot obtain that for specific households, for instance. But similarly, I can say that if you're interested as a as a legal authority in specific use of drugs of abuse, for instance, in prisons, you can try to sample the outflow from the prison itself in order to find data about how much is used in such a penitentiary. Okay, I think I will finish by acknowledging some people, the colleagues from the European City Monitoring, of course my colleagues here in the Netherlands for this work. We were supported by the Dutch research program of the water companies and by the European Monitoring Agency for Drugs and Drugs Addiction.